Okay, so whenever a new virus pops up, the world kicks into this frantic race, right? A race to figure it out, a race to treat it, and a race to make a vaccine for it. But what if we could build a toolkit that's ready to go before the next pandemic even starts? So today, let's dive into some really cool new research that explores this idea, using tiny, programmable molecules to get ahead of the game. Here's the fundamental problem we're always up against: viruses. And I'm talking especially about RNA viruses like SARS-CoV-2. They are absolute masters of disguise. They mutate all the time. And that means a treatment or a vaccine that works great today might not be so hot against a new variant tomorrow. It's this constant biological arms race. And frankly, we need smarter weapons. So, how does a virus actually infect us? Well, think of it like a burglar. Our cells have a door with a very specific lock on it. In this case, it's a receptor called ACE2. The virus has the perfect key. That's its spike protein. When that key fits into that lock, the door swings open and the virus gets inside to make copies of itself. This moment right here is critical. The entire goal for a lot of antiviral drugs is to stop that key from ever turning in the lock. The solution this research looks at comes from peptides, which are, the basic building blocks of life itself. And we're going to look at two really clever strategies. First, a direct therapeutic design to block the virus, and second, a platform for a whole new way of making vaccines. So what exactly is a peptide? The simplest way to think about it is this. If proteins are like long, complex necklaces, peptides are just very short chains of the same beads, which are called amino acids. The real beauty of peptides, their superpower, is that they're so specific. Because they're small and you can design them, scientists can basically program them to do one very particular job and do it really, really well. All right, let's jump into strategy number 1, the decoy key. The idea here is brilliant because it's so simple. If the virus has a key, how can we design a peptide to basically jam the lock before the virus even gets there? And this picture shows you exactly how it works. On the left, there's the virus with its spike protein key. Normally, it wants to connect with the ACE2 receptor on our cell. But here comes our little hero, the peptide inhibitor. It's designed to be a perfect decoy for that key. It latches right onto the virus's spike protein, effectively gumming up the works. Now that the virus's key is busy, it can't unlock our cells. It's totally neutralized. So the researchers tested 2 different versions of this decoy. The first one, they called P1, was basically a direct copy of the part of our own ACE2 receptor that the virus likes to bind to. Pretty straightforward. But then they made P25, and this was the upgraded model, specifically engineered with mutations to make it grab onto the virus's spike protein even more tightly. And this right here is the million-dollar question. I mean, it's one thing to design something that works in a perfect lab setting, but how does it do against the virus as it actually exists out in the real world? Could this souped-up peptide, P25, handle not just the original virus, but that notoriously tricky Omicron variant? Well, the results were absolutely stunning. This number you see, 60.8 micromolar, that's the IC50 value. Just think of it like a golf score for potency. It's how much of the peptide you need to block half the virus. And just like in golf, a lower score is dramatically better. Against the original and Delta variants, the score was way up around 450 to 550. Against Omicron, it was just 60.8. The engineered peptide didn't just work, it was almost 10 times more potent against the newer, tougher variant. Just wow. And if you're a see it to believe it kind of person, this slide is for you. What you're looking at here, each green dot is a cell that's been infected by a pseudovirus. It's a safe lab version of the virus. In the control rows up top, you see tons of green. That's bad, lots of infection. But just look at the bottom row for P25. As the scientists add more and more of the peptide, poof, the green glow almost completely vanishes. That is the decoy key in action, stopping the virus dead in its tracks. Okay, so that decoy key is an amazing therapeutic for treating an infection. But what about prevention? Well, that brings us to the second strategy, using these same peptide building blocks to create a smarter type of vaccine. The goal of any vaccine is to train your immune system, and this platform aims to create the perfect training dummy. It's a synthetic particle that, to your immune cells, looks and feels almost exactly like a real virus. It shows them precisely what they need to hunt for, but without any of the actual danger. So how do you build this little training dummy? The process is so elegant, it's almost like magic. Scientists start with these custom peptides that have a special property. One end loves water, the other end hates it. They just dissolve them, let the solvent evaporate, and then add water. And that's it. The peptides do all the heavy lifting themselves, spontaneously snapping together into these perfect, tiny little spheres. So why go to all the trouble of building these spheres? They call them peptide amphiphilic micelles, or PAMs for short. And this slide lays it out perfectly. On their own, floating around freely, peptides are kind of fragile. Our bodies break them down pretty quick. They also don't really get our immune system very fired up. But once you assemble them into a micelle, they become stable, they're protected, and their virus-like shape basically acts like a giant red flag for our immune system, triggering a much, much stronger response. So when you put it all together, the advantages of this PAM platform are just crystal clear. These things are stable, they protect the important bits, they're incredibly safe, they showed almost no toxicity in the lab tests, and most importantly, they do a fantastic job of activating the key players in our immune system, and they do it because their tiny spherical shape is the perfect mimic of a real viral threat. All right, let's zoom out. We've seen a powerful therapeutic decoy and a really promising new vaccine platform. But what does all of this mean for the big picture? For fighting the pandemics of the future? This right here is the key takeaway. The true power of this peptide technology isn't that it's just a solution for one virus. It's a platform. Because you can design and synthesize new peptides so quickly, you can adapt this whole toolkit on the fly. When a new virus, let's call it disease X, shows up, you could theoretically design, produce, and start testing new peptide decoys and new vaccine micelles in record time. And hey, you don't have to take my word for it. This quote from the research abstract says it all. This isn't just some neat science experiment. It's a promising and scalable strategy. The ability to address not just the threats we have now, but future viral threats. That's the holy grail of pandemic preparedness. So it really leaves us with this truly exciting thought. We often think of fighting pandemics with these massive global efforts. And of course, those are absolutely essential. But this research shows that a huge part of the solution might be microscopic. These tiny, elegant, self-assembling molecules that could finally give us the adaptable tools we need to get ahead and stay ahead of the next great viral challenge.